Using OHS and other population cohorts, we were able to demonstrate that ASE may be contributing to variation in aging processes. Hi, I'm Michelle Harwood, a recent PhD graduate from the University of Toronto and the Ontario Institute for Cancer Research, and I studied under the supervision of Dr. Philip Awadallah. I was interested in the research question, why do some individuals age and remain healthy, whereas others are going to progress to disease earlier on? Over the next few minutes, I'm going to share some of the research from my PhD, which addressed this question using Ontario Health Study data. And I showed that allele-specific expression can actually contribute to some of this variation we see in healthy versus unhealthy agers. We know that human populations are very diverse in phenotypes, such as who develops a disease and the severity of that disease. But there is still a long-standing investigation into why some individuals who have the same genotype, the same genetic makeup, still have such highly variable phenotypes. We can use a population genetics lens here to start to investigate what is going on between the genotype to phenotype that is causing some of this variation. In my research, I used a unique data set from the OHS to show that transcriptomic differences between individuals may contribute to some of this variation we see in healthy versus unhealthy agers. This research project focused on the phenomenon called allele-specific expression, or ASE. And this is where at a particular position within an individual, you may have unequal expression of the two alleles. And this is important in a disease context because let's say one individual, such as the top example here, has a disease-associated allele that is being overexpressed. This individual therefore may have a high risk of this disease. Whereas the second individual on the bottom, which is underexpressing this exact same mutation, may be at lower risk of disease because of that underexpressing ASE. There are many phenotypes and diseases that can be associated to ASE, many of which have already been looked at in the literature. However, I was interested in this aging question to see if ASE is associated with any variation in healthy versus unhealthy agers. We leveraged the Ontario Health Study data, specifically a data set that was uniquely selected based on these age-related questions. Dr. Alyssa Bader, a recent graduate from the Awadallah lab, selected participants based on a risk score using complete blood count data. A low risk score essentially means low risk for mortality. So using this risk score in combination with age allowed Alyssa Bader to look at an extreme sampling approach so we get a unique set of individuals that are young healthy, young unhealthy, aged healthy, and aged unhealthy. From these individuals, single-cell RNA sequencing was performed and 22 unique cell populations were identified. For the ASE study, we grouped these cell types based on their involvement in adaptive versus innate immunity. We showed that there is an increase in ASE with age, but that this was specifically observed in those cell types involved in adaptive immunity. Now, when we separate participants based on that risk score, low risk versus high risk, we see that low risk individuals actually have a larger increase in ASE with age compared to high risk individuals. Again, this was specifically observed in the adaptive immune cells. We also identified this same pattern in bulk data from Cardigen CanPass Quebec cohort. This research leveraged this unique data set using single-cell transcriptomic data from the Ontario Health Study to identify that ASE may be contributing to some of the variation we see in healthy versus unhealthy agers. Other research not shown in this presentation include that many of these patterns may be driven by immune genes. We also identified that individuals with disease, such as type 2 diabetes, demonstrate opposite patterns to what was demonstrated here. Lastly, we also showed association with medication, where individuals taking medication have larger increases in ASE compared to individuals not taking medication. Using OHS and other population cohorts, we were able to demonstrate that ASE may be contributing to variation in aging processes, such as healthy versus unhealthy agers. And this research could be important in the future for biomarker development and precision medicine studies.